Hey all, welcome to ShareTreck. Today is the Sunday edition, 8th of May 2022. And today I want to talk to you about Omics. Many of our subscribers are already well versed with genomics, and you've already been dealing with shares. But there might be some of the subscribers who are new to this field. And I thought that when I started looking into genomic shares, the first thing I did was to look around and gather enough information about this field. And the collection of all that information is going to be presented in this video as the precursor to understanding omics. So with that said, let's get started. Before we get started, I just want to give you a brief background about myself. I have a bachelor's in commerce and an MBA in information technology. And throughout my career, I have been working in information technology business. And whatever I'm talking to you about biology out here is all things that I have picked up over a period of time. And of course, my high school biology became the foundation for that. But most of the things I've picked up is from the internet. In order to understand my investments in genomics, I had to do this. And what I'm going to talk to you today is going to be about the building blocks of knowledge that I accumulated. This is the first part of the video, as I said. And also I wanted to say a few more things before I proceed. The first thing is that I'm trying to keep most of these videos under 20 minutes because that's the feedback I have got from the subscriber base. The other feedback I have from the subscriber base is that they need more depth, and I'm working on that, but giving more depth in 20 minutes seems to be a tall order, especially for a complicated subject like genomics. So what I have done is yesterday I dropped a video, which is my first experiment, where I have zoomed in on CRISPR and ViaCyte VCTX210. And I want your feedback. Do you consider the duration of the video to be adequate? And do you consider the depth to be adequate? Please put your comments below. It will help me to calibrate and make better videos that are more suited to your time availability, as well as the depth that you desire to see in the topics. The other thing I have also found after being able to put videos every day consistently for almost 4 to 5 months now, is that there's a bit of a burnout that happens when a video comes daily. And also there's a compromise in the quality of the video because a good video would typically take 3 or 4 days to make. For example, the CRISPR video that I put yesterday, it took me 2 days to make. I had been working on it for almost 2 or 3 days, and then I was able to upload it and get it ready. So to do a quality video in a short duration with greater depth, it needs more research. So I'm thinking of working around and trying to reduce the duration of the daily update and work on condensing the technical analysis portion, because right now technical analysis doesn't make much of a sense for the genomic stocks. I would like your opinion on that. Please put your comments below. I want to try to make sure that the daily update doesn't exceed 10 to 12 minutes. And that way, what I would like to do is for the rest of the day, I would like to take some time to come up with a much more comprehensive and shorter video for the end of the week. I'd like your feedback on whether I'm going in the right direction or not. So with that said, let's get started with our building blocks for genomics in order to understand omics companies. The first thing I'd like to do is to introduce some of these terms: DNA, genome, nucleotide, gene, mRNA, tRNA. So this is going to be a kind of stream of consciousness information, the way I had gathered it. So I'm going to talk about these terms because we need to understand these terms in the same way so that we can talk about the omics companies. To begin with, the DNA is become very famous because it's there in popular fiction, like CSI. In the crime scene, the detectives go and they say they want to collect a DNA sample and so on. So DNA has become much more popular. And people understand the term to mean something to do with identifying a person. But technically speaking, DNA is deoxyribonucleic acid, and it's a molecule that contains biological instructions that make each species unique. A fish has a unique DNA, humans have unique DNA, and snake has a unique DNA, and so on. DNA, along with the instruction it contains, is passed from adult organisms to their offspring during reproduction, and some traits from one of the parent and some trait from the other parent combines and expresses itself in the offspring, and some defects from the parent's genes remain dormant in the offspring's genes, and some defects from the parent's gene become expressed in the offspring's gene. Therefore, you might find that as the human race multiplies, or as a batch of fish multiplies, you will see variations in them. All those variations are coming from the expressed part of the DNA and unexpressed part of the DNA. So during DNA replication, DNA unwinds so that it can be copied. In its wound up form, it's called the gene. At other times in the cell cycle, DNA also unwinds so that its instructions can be used to make proteins and for other biological processes. But during cell division, DNA is in its compact chromosome form to enable transfer to new cells. Researchers refer to DNA found in the cell's nucleus as nuclear DNA. An organism's complete set of nuclear DNA is called its genome. Another fun fact is that the term genome is derived from gene and chromosome, hence genome. Therefore, genomics deals with the entirety of an organism's hereditary information coded in its DNA. And DNA is made of chemical building blocks called nucleotides. And nucleotide is where you have those block alphabets that you see in popular literature, where they talk about genes. You see T, A, G, those kind of block letter alphabets. They are coming from nucleotides. Nucleotides consist of phosphate group, a sugar group, and one of three of nitrogen bases. And those nitrogen bases are adenine. So you have A from adenine, T from thiamine, and G from guanine, or C from cytosine. So those are the block alphabets that go into forming the DNA. And the sequence in which they are stringed together determines certain traits, like whether a child is going to have blonde hair, or it's going to have brown hair, whether it's going to have blue eyes or black eyes. Those kind of things are coming through the combination of these block letter characters or nucleotides, the sequence in which they are organized. The human genome consists of about 3 billion base pairs and 20,000 genes and 23 pairs of chromosomes. That's what is the scope of what these companies are trying to work on. The Omics companies are working on this huge range of information. Having said that, I would like to bring the analogy of something from the technology field. We have object-oriented programming, where there are objects, and each object responds to a message that it gets. And how it does that is by having data stored inside it, as well as instructions to operate on the data. So it more or less behaves like an organism. This is the closest parallel I can draw to from my native field of information technology to what we have here in genetics. The same way, you can imagine the DNA to be a database of information, a record of things, and some of the information is for synthesizing proteins, some of the information is for expressing traits in the offspring, and so on. And DNA contains the instruction needed for an organism to develop, survive, and reproduce. In order to do that, it has to synthesize protein, it has to metabolize energy, and so on. Each DNA sequence that contains instructions to make a protein is known as a gene. So DNA's instructions are used to make proteins in a two-step process. First, the enzymes read the information in a DNA molecule and transcribe it into an intermediary molecule called as messenger RNA. The process in which the genetic code in the mRNA is read to make a protein is called translation. This is done by the ribosome, and tRNA helps to bring amino acids in the right sequence to assemble the protein. So tRNA is more like an engine. I don't know how many of you have seen a railway yard where the bogies are arranged for a compartment for a train. So let us examine a goods train. There is an engine and there are multiple compartments behind it. So the engine driver gets the instructions which are the compartments that are to be picked up and in what sequence they have to form behind the engine. So he goes from one track to another and people connect the compartments in the sequence, and then finally the train is ready to go. So tRNA is like the engine driver. It understands from the mRNA what is needed. It goes and picks up the respective amino acids, brings it back and put it in the right sequence so that a protein is formed. So that's how things work. So now let us examine the major omics. There are 4 of them. So the first one is genomics. It explains what can happen. The second one is transcriptomics. Transcriptomics deals with the entirety of RNA transcribed from DNA. So that's the area of expertise out there. Proteomics explains what makes it happen. It deals entirely with the proteins translated from the mRNA, the proteome. And metabolomics explains what has happened. And there is another omics called as epigenomics, which addresses factors and mechanisms affecting the accessibility of genomic information by modification of its structure via DNA. So genomics can be summarized as the mapping of all human genes and their interactions with each other. And genomics provides a static view, the DNA, which is the blueprint. The other omics are dynamic and subject to change under different conditions. So there are companies which deal with each of these areas and also in creating tools that help companies work in each of these areas. And that is the scope of our investment when we look at omics companies. So the domains of expertise, if I was to talk about them, is sequencing genomes, that is one domain, compiling database of mutations and describing what each of those mutations does is another area, because if there's a library of such mutations and their effects, then that can be used by companies doing therapies to identify and edit genes and correct those mutations. Compiling database of proteins and their role in biological system. So there are some bad proteins that cause nerve damage and conditions like that. So the database of proteins and their role in the biological system, and which sequence of genes are responsible for producing those proteins. Once that kind of information is available, then companies can go and create therapies which can correct defects and make sure that the bad protein is not created and therefore solve the ailment. Synthesis of genes, editing genes to create therapies, editing genes to impart properties, that is more in terms of in agricultural practice or horticulture, where you want to create crops which are drought resistant, or plants which give more fruits, or things like that. You get the idea. And creating hardware and software system to help all of the above to be done is another field. You have companies which we talked about. We spoke about Illumina, PACB. They are manufacturing devices which can do sequencing, long reads and short reads. So those are all tools that can be used. Of course, you understand CRISPR-Cas9, Cas11, Cas13, smaller proteins, the smart protein, smart editors. So all those things are available. And many of the companies that we have already seen and have in our watch list are involved in most of the enterprises out here. I'm going to give you some sample companies which operate in each of the omics that we talked about today. I'm not going to talk about genomics, but I'm going to talk about transcriptomics, proteomics, as well as metabolomics. So let us start off with transcriptomics companies. 10x Genomics is one of the biggest that I can think of. It is in it's got a ticker symbol TXG. You can find it in NASDAQ. The last I saw, it was quoting at $51.19. Fluidigem is with a ticker symbol FLB. It's in the European bourses. It costs euro dollar 2.68 as of market close on Friday. Akoya Biosciences, AKYA, $9.73. Nanostring Technologies, NSTG, $17.20. So these are in NASDAQ, and these are all sample of companies that operate in the transcriptomics area. And next, if we look at the companies which are in proteomics area, the first is Proteomics International Labs. It's Australian dollar 1, and ticker symbol is PIQ, and that is in Australian bourses. And Persis Proteomics Inc., which has got a ticker number 4882. It's the Japanese exchange. In Japanese exchange, they have ticker numbers instead of alphabets. JPY 370, that is Japanese yen 370, is the price for the share as of Friday. SomaLogic is another company which is quoted in NASDAQ, $6.21. SLGC is the ticker symbol. And if I'm not mistaken, I think SomaLogic has got something to do with Illumina or CRISPR Therapeutics, one of the two. Biognosys is a privately held company, so there is no ticker symbol. These are all the companies that are operating in proteomics areas. And the reason I'm showing this to you is basically because you can do your own Google search and find more information about these companies. And wherever available, I have given you ticker symbols so you can go to your trading platform and get more information about these companies. And one of these days, when we have time, I would cover some of these companies in a Saturday special or a Sunday special kind of a video. And next, going into metabolomics companies, you got a Metabolon, which is a privately held company, so we don't have a ticker symbol. Agilent Technology is another big one. Symbol is A, very classy. It's going to be on the top of the list. Its last quoted price was $121.08. Then you got Human Metabolome Technology Inc. This is quoted in the Japanese exchange at 6090 being its ticker symbol, and it's at 570 Japanese yen as of close of Friday. Biorad Laboratories Inc. With a ticker symbol of Bio is on NASDAQ, last quoted at $501.96. So this brings us to the end of this video today. What I have tried to do is use half of the video to communicate with you about my own queries about what we are doing with this channel. Are we going in the right direction? Do you like how subsequent videos are coming up? I need some feedback from you, good as well as bad. If it's bad feedback, please keep it respectful so that it doesn't demotivate me too much. And it's very important that we get everything right so that our channel is much more useful for all of us. And I'm looking forward to getting more subscribers here so that once we touch 500, we can have a community tab and have much more two-way collaboration. Right now, it's a one-way. I'm putting things out there, and when nothing comes back to me, I don't know what's happening. If you give me some feedback, I can always work on that and try to improve things. Otherwise, the improvement is going to be slow and may or may not reflect what you're looking for. So with that said, please put a like on this video if you liked it. And if you're not subscribed, please subscribe and become a member of our community. We're going to talk about everything CRISPR and Omics, and also about stocks in general, technical analysis, and how to identify the trend in the market. And friends, we are going through a very, very difficult time. Please be careful with your investments. It's a very volatile time. There is no bull market yet. We need a lot of risks to be resolved. And the Fed has got to bring the inflation under control. The recent employment report has not brought any kind of dovishness to the Fed. Instead, it has shown that the economy is red hot. So a little bit of cooling is needed. So the Fed is going to be doing what it needs to do, increase the interest rates. There's a potential recession in Europe because they're going to put an oil embargo in place with Russia. So be very careful, be patient. All these bad times won't last. In the long term, all the genomic companies that we see here are going to be good bets as long as they have got a healthy balance sheet and good pipeline items which are very close to getting a BLA. And that's what I would say. So with that, I would like to end this session. Bye for now. I'll see you on Monday.